Yeah, good morning, and thanks, Peter. Uh, as a matter of fact, I'm pretty excited about this morning. And this is actually towards a cancer-free world. It's something I was wondering for a while. And I hope at the end of my talk, you probably understand where I come from. Um, so this is overall slide learned from the leader and Dr. Gleaf, and really trying to say, hey, what you're trying to do, can you, generate a good knowledge? If possible, can you really translate certain of your discovery into application? And this slide basically, summarized the major activity happened in the lab. As you can see, the project line between the discover and also between the product. So this is called translational. In the center, we also have a call in the lab. So today, I am going to share a couple of stories and how we pick up a research topic and follow through, and also what are the criteria we use to pick up a specific kind of research project, and what eventually we actually heading to. So the first story is about NEPC. So I want to ask a question on behalf of you, why you focus on NEPC. So NEPC is the subtype of prostate cancer. And in fact, the de novo NEPC is very uncommon. About 10 years ago, if you're working on NEPC, actually that would be a very bad idea, because that's not a major subtype in the field of prostate cancer. But during the last few years, 5-6 years, and because the most potent AR pathway inhibitors are used, people found that the treatment-induced NEPC incidence is a significant increase. It's up to 17 to 25%. And NEPC is no effective therapy. So you can see this disease, kind of could be back to 10 years ago, could be a new next generation prostate cancer disease, and there is no particular good therapy, and was not hot. Those are the criteria we use. We say this is a good, we need to work on this one. Particularly, you don't have anything to study for the transdifferentiation from adenocarcinoma to NEPC. So another criteria we use, if you come to the field and it's obviously it's hard for you and for everybody, can you have a chance to be better? And so the answer most likely is yes. So in order to study this transdifferentiation, the first things you want to model the disease, and then based on the model, you can do the discovery and follow through. If any discovery you have, done in the artificial model, you want to make sure they are relevant to the disease space you're working on, and then follow through with functional studies and mechanism, if possible, something more than that. And this is back to 10 years ago. And when we get a piece of adenocarcinoma from Martin's surgery, and she actually established a hormonal naive prostate cancer model. And this is really cool because, for hormonal naive prostate cancer, it's known to be very hard to grow in mice, but we managed to grow them and establishing LTL-331. Because this model is a hormonal naive one, that gave us chance to mimic the clinical progression. So we did a whole, host castration to mimic androgen ablation in the clinic. And really interesting. So initially, both tumor volume and the PSA coming down to the bottom. And after a few months, and tumor coming back, but PSA stay as undetectable. So this is really interesting because you may wondering whether or not you still have the recurring tumor is still human, still prostate, and if all yes, and what kind of prostate cancer you still get. So after all those kind of examination, we found, that the recurring tumor is not like adenocarcinoma anymore, but was kind of a typical neuroendocrine prostate cancer. So of course that was too new back to 10 years ago. And I remember I talked to Martin. He basically, and he did not believe that. He probably think this is a kind of artificial system anyway. So we have to really work hard, repeat this experiment 3 times. And each time it take about a year to finish. And every time we got a very consistent NEPC to coming back. And then we were convinced. And one day, and Martin really called me, and he said, YZ, come to my office. And I said, can you talk to me over the phone? He said, you better come to my office. I have a good news to share with you. And his good news actually is the bad news for the patient. So the patient who donate the tumor actually from adenocarcinoma also recurring as an NEPC. So that's time actually we pay more attention to this neuroendocrine transdifferentiation. Of course, then it was excited, but still kind of new to the field. And this is a kind of histopathology, and you can see how the morphology change during this transdifferentiation. So initially, you get a typical glandular structure, androgen receptor, PSA, all positive, and then the AR-PSA coming down, and then the NEPC markers show up. So this is a kind of a model really gave you a very unique, actually even up to today, this is the only PDX model show this transdifferentiation. So if you don't have this model, it's hard for you to study anything in between. You can compare NEPC with adenocarcinoma and find some specific transcription factors and so on and so forth. But you don't have a chance to study early contributors during this process. So again, this model gave you a time series, and then you can follow this time course to ask a good question. What are the factors drive the NEPC development? Of course, and we want to look at a big picture and from two aspects, either from a genomic or epigenetic genomic aspect. And in collaboration with my dear friends Colin, and he used all the weapon he has, and using a re-CGH, a bulk sequencing, and later recently have a single cell sequencing. And in this particular model, he couldn't identify any significant alterations between the 331 and 331R during this transdifferentiation. So this is really gave us a hard time. If we find any kind of mutation or other genomic alteration, that would be easy to follow. But his data suggests the genomic alteration may not be a major contributor for this transdifferentiation. That actually led to our new hypothesis. Then the new hypothesis may suggest us to focus on epigenetic alteration during this process. We're also trying to focus on some early driver, means it's not something only show in the terminal NEPC, but show up earlier and consistently upregulated. And if functionally important, they could be therapeutic target. That was the hypothesis. Of course, I Dong and Xinpei in the lab using multiple filters and narrow down a gene called actually a protein is HP1 alpha. The gene called CBX5 is one of the epigenetic reader. And so it's very interesting, they further identify 36 heterochromatin genes as a signature for NEPC. Within those heterochromatin genes, HP1 alpha is really the one and show up earlier and continually up-regulated in terminal NEPC. So this is in the RNA level, you can see the protein, and even we look at IHC. So, and this phenomena also demonstrate in cell line and also multiple NEPC PDX models. So HP1 alpha, as I mentioned earlier, this is a short form for heterochromatin protein. So we know the heterochromatin signature is a typical one for NEPC. How about this gene, CBX5? CBX5 actually can recognize the histomarker H3K9 trimethylation. So this is a histomark that can really regulate gene expression, particular repress gene transcription. So this is the gene certainly can affect many other gene expressions if the gene functional. So the next question we ask, and is this the finding is also kind of relevant in clinical NEPC? Otherwise, you have no reason to follow through. So we use back to years ago, Xinpei used multiple clinical cohort and demonstrate indeed HP1 alpha is significantly upregulated in patient NEPC. And recently, don't use the other latest two bigger clinical cohort also prove this is still the true case. So all the available cohort suggest the HP1 alpha is relevant to patient NEPC. This actually gave us confidence to do further study, and in terms of functional study, and traditionally, this is the kind of people tell you. I don't have to go to the detail how to describe this gene. So this is a functional study, and Xinpei using both cell line, this adenocarcinoma cell line, and particularly LTL331 organoid model. She overexpresses HP1 alpha, and she can increase the neuroendocrine marker expression. And of course, the marker expression is just the surrogate parameters, and so it's not too much. And she also knocking down the gene in NEPC cell line. What she can find is the cell viability, cell numbers, colony formation, DNA synthesis, whatever you can thinking about it. The conclusion is when you're knocking down this gene, actually you inhibit NEPC cell growth. And he actually detected, the induced cell growth inhibition is due to NEPC cell apoptosis and cell death. So I don't have to go to all the detail. So if you have a cell that slow growing already, you put in the animal, of course they show smaller tumor. So this is a kind of a declaration from in vitro to in vivo, but it just tell you, knocking down this particular gene really affect tumor growth. So as a scientist, people always trying to say, hey, we have to know how it works. So this is so-called mechanism study. As I mentioned earlier, and HP1 alpha is known to recognize a histone marker, H3K9 trimethylation. So, and Xinpei did colocalization HP1 alpha with H3K9, and she found that they truly correlated with each other in NEPC, but not in adenocarcinoma. And he also found that HP1 alpha and enriched the histone mark on androgen receptor and REST promoter. And then actually when you overexpress HP1 alpha, you suppress both AR and REST expression. That's actually is a big deal. The reason it is a big deal because there's a AR and a REST are typical, kind of adenocarcinoma transcription factor. So in order to transdifferentiate into something else, you got to shut down AR and REST. And so this is actually the data here. You just see in NEPC, the H3K9 and increased in AR and REST promoter region. So that's actually the data from chip sequencing and chip PCR data. So this is the kind of really a brief summary to say when HP1 alpha increase and you enrich H3K9 trimethylation and in particular on the promoter region of AR REST. So that suppression AR REST expression that could be really fundamental for the transdifferentiation. And moreover, and she also found this is kind of knocking down HP1 alpha in, really affect multiple cell cycle and DNA damage related pathway. So that result suggests, the HP1 alpha also important for the NEPC progression. So of course, there are more data over there, and I just want to give you a short conclusion here. So after all those studies, you discover a new players. This is HP1 alpha is relevant to the clinical NEPC, seems functionally pretty important. We sort of understand how it works. So we're wondering, can we translate this knowledge into a drug development? So this is the kind of direction we're heading to, again, in collaboration with Art and Michael, and we're trying to target the Chrome domain of HP1 alpha. So the reason we want to target that is kind of this pocket is the place for HP1 alpha to recognize the H3K9 trimethylation. So we hope we can develop a small molecule and interrupt the interaction between HP1 alpha and the histone mark. So this is the kind of a long story. I just want to stop here and switch gear a little bit. So we want to, and there are many of those potential cancer-related genes. And as a matter of fact, most recently there is a paper document. And, if you talk about a cancer-related gene, that probably majority human gene has been reported to one way or another, reported by literature. They are related to cancer. And that's actually kind of scary too, because you have, cancer are so challenging. You have so many genes, and nowadays not only just 25,000 coding genes, you have non-coding genes as well. So that it's really when you zoom out to see the whole picture, we are not winning the war on cancer, although we feel we should be able to do better job. But come to the advanced cancer, no matter how smart we are, we eventually, most of the cases, we're still losing the war. And one of the excuses all the cancer researcher like myself has, because cancer is too complicated. And this is not because we're not smart enough, because cancer is smarter somehow. And for this kind of excuse, we feel okay. Again, back to exactly 10 years ago, when I got a smart student come to me, which is Stephen Cho. So I realized I'm not going to give him a very easy, just one gene, and then follow through to have a few publication and a graduation. And so I want to challenge him. I said, okay, why not you go back to visit the fundamental of the biology, cancer biology, the so-called fundamental. And the first paper you want to read is a hallmark of cancer, published by Anahan and Winneberg. And then they actually, at that time, they just published the update version in 2011. The number of hallmarker increased from original 6 to 10. I asked the students in the lab, what would be the number in 2020, actually, if they could come up with another updated hallmark of cancer review article. And we were convinced if that would be the case, the number could be more than 15 or 20. And of course, why is that? Because the knowledge kept increasing and the technology actually became more advanced. And then we look at the situation. It seems the trend for cancer researchers, and not only cancer researcher, and it seems that each one find a new subtype of cancer or molecular subtype or small whatever genes, and you become a number one in what you are studying. And so the question we're trying to ask, so we're trying to going up like this tree demonstrate here. And, we know, for example, prostate cancer, probably we know this is just a prostate cancer. Then later on, we know prostate cancer, adenocarcinoma, and neuroendocrine carcinoma. Now we know even you have a double negative, you have a submolecular type, which could be very useful. But one thing we are missing, and for all the cancer, what is the most common ones? And that could be more fundamental to not only just the subtype of cancer, but applicable to all cancers. And so the real question is, are we really going up or going down? Or should we consider both directions in our life? So, and I gave Stephen some hard time. I said, okay, you need to look at the hallmark of cancer at a different angle and find out what are the unique and fundamental hallmarkers and to cancer. It's not just like we focus on angiogenesis, proliferation, apoptosis, all kind of metastasis. Metastasis actually is a name for cancer, but our lymphocyte can move around naturally in our body, but we don't call them metastasis. And proliferation pretty often happen in normal tissue as well. So one of the angles we're trying to look at back to before immunotherapy became so hard, and we often heard cancer cell pretty often living in lymph node, just like a criminal living in police station. And so why cancer can live in the police station? So we actually trying to say if a criminal can living in a police station, they need to wear their uniform, need to speak their language. So based on that kind of assumption, and also the literature search, Stephen came up with a hypothesis called epithelial immune cell transition. So of course, what's the significance of this hypothesis, and what's the use for this hypothesis? This may just give you a concept to say, okay, cancer, if you get a successful cancer cell should know how to communicate with the immune cells. And but it was very challenging, we know, and there are multiple types of immune cells. There are multiple mechanisms. Here, just want to show you the T cell. T cell, they throw multiple, kind of immune regulators to interact with cancer cell. A very famous one we all know is a PD-L1, PD-1, right? And but a PD-L1, PD-1 is just one of so many, players between T cell and the cancer cell. Imagine you have NK cell, you have B cell, you have macrophage, and there's so many different type of immune cells. So then we ask next question, and it's what would be, the major communication code between cancer cell and the immune cells. And so again, back to multiple years ago, we look at all solid cancer. There are two phenomena associated with all solid cancer, which is it seems all tumor microenvironment is immune suppressive. We could call cancer as a localized immune suppressive disease. Another phenomenon is the tumor microenvironment is always acidic. And those two phenomena are best known for years. And the real question is there any connection between acidic and immune suppressive tumor microenvironment? So again, this is the Stephen Chow. He was very brave. And he came up with this hypothesis and back 2 years ago to say, hey, actually, the acidic microenvironment probably is responsible, at least partially, for immune suppressive immune suppression of the anti-cancer immunity. So, and actually, the acidic microenvironment mainly caused by lactic acid generated by cancer. So cancer is a major player that's responsible for their own microenvironment. So back to 8 years ago, that was a new hypothesis. And just recently, Joy and the team came up with a review article and document all those innate and adaptive immune cells, all affected by the acidic microenvironment and lactic acid. So they are multiple mechanisms. It's a kind of interesting. You have more lactic acid in tumor microenvironment that will suppress immune cell proliferation. And if the lactic acid concentration is too higher intracellularly, that can induce cell death like apoptosis. And lactate also can function like epigenetic regulator and to changing the gene expression. In this case, they can induce macrophage dedifferentiation and by convert macrophage type 1 to macrophage type 2. And of course, lactate also can function as function like signaling molecule, induce antigen presentation suppression. So they are new mechanisms. So in our field, in our lab, and Stephen actually identifying the lactic acid actually transported by a gene called MCT4. So that's actually the MCT4. It seems relevant to multiple cancers. Here we only reported in prostate cancer. So I want to show you a small kind of video that Joy made.
Our bodies are made up of trillions of cells. Just based on the sheer quantity alone, the probability of there being mutations is likely. But when there are only one to a few cells that are abnormal or cancerous, our body's immune system is on standby. And in this early phase, the cancer is under control. In order to survive and grow, cancer cells generate a lot of building blocks using an altered metabolism. The process is called glycolysis and fermentation. A byproduct called lactic acid is discharged out of the cell. Lactic acid pools around the cancer cells, making the microenvironment around the tumor acidic. This is one of the main methods cancer cells use to suppress anti-cancer immunity. It drastically reduces the survivability and growth of surrounding immune cells. Not only that, it also causes loss of their functions, further allowing for cancer's continual growth. The acidic microenvironment has other effects as well. It also stimulates the growth of blood vessels through a process called angiogenesis. This promotes the spread of cancer locally and systemically. The acidity will also decrease the affinity of oxygen binding to hemoglobin, lowering oxygen levels in the area. This results in a toxic environment, a hallmark of aggressive cancers. So what's the most effective way to target these fundamental characteristics of cancer biology? The strategy of LAST is to develop a small molecule inhibitor that blocks the lactic acid transporter called MCT4. Successful inhibition will result in two main consequences. Firstly, cancer cells cannot get rid of their lactic acid byproduct. The accumulation of acid within the cell would slow down glycolysis, prevent cell growth, and directly kill the cell. Indirectly, as the acidity in the surrounding environment reduces to almost normal levels, lactic acid's effects of hypoxia and angiogenesis will begin to revert. Most importantly, those anti-cancer immune functions that were inhibited earlier will be regained. Immune cells can start attacking cancer cells efficiently again. Thus, MCT4 inhibition via LAST will not only kill cancer cells, but also restore physiological conditions to recover anti-cancer immune function. This could be one of the most powerful immune therapies that's widely applicable to most human cancers. Our bodies are made of
So the idea is really just we're not trying to inhibit the generation of lactate, but blocking the transporter, and then cause the direct killing and indirect killing. That would be the main thing. So again, in collaboration with Art and Michael, using their powerful and artificial intelligence in silicon program, and we screen for MCT4 inhibitor. So this is a kind of pipeline they used, and we already gone through 10 rounds of those, and bought about 500 of the small molecule. And so once we get a small molecule inhibitor, actually the small molecule and will be tested in the lab using multiple assays in vitro, and also eventually to the in vivo toxicity and efficacy evaluation. So this, we got a marvelous group of people, and Stephen, Jennifer, Fiona, and Connie. So they have their kind of a different expertise in what they are doing. So I just want to give a little bit of flavor here, and so this is the kind of culture system and Jennifer and Connie used. And when you see when you only have either NK cell or compound alone, you can partially inhibit tumor cell growth. But once you combine them together, and they basically eliminate all the cancer, that's basically suggests that our compound actually not only prove our hypothesis, actually is really effective when you have immune cell in place. So this is a co-culture system they established and use T cell, actually the active PBMC cells. And with the compound here is 448. Again, when you have the compound alone or PBMC alone, so PBMC in this case have a minimal effect. But when you have the two together, you can see the cancer cell actually significantly reduced. So I just want to, there are many exciting data, but what I'm trying to give you the concept here is this is the kind of, we come up with a working hypothesis. Again, we follow through the discovery, the clinical relevance, and now come to the drug development and trying to now even we have establishing a company and which come back to support the drug development. Anyway, so I want to conclude with another topic a little bit. And nowadays we focus on tumor dormancy quite heavily, actually for it's at least 7 years, but we haven't really published too much. So tumor dormancy actually was a very interesting thing because back to 5-6 years ago, when Francisco was here, he came out with a hypothesis. What he noticed that, and the more aggressive cancer you have, and theoretically, they probably have a more genomic or epigenetic alterations. Actually, the less dormancy capability they have. And that's why leading to a hypothesis called this dormancy, tumor dormancy is a non-genomic disease. Perhaps it's an epigenetic regulated things. So I use this one, just want to share some of the principle we use, why we want to study tumor dormancy. And the 3 criteria are, this is a critical issues you want to study. And for dormancy, which is very critical because dormancy is the one give you recurrence and the metastasis. And so we don't know how to manage that. So this is a critical question in cancer field. And the second, and this is already hot or still kind of in their early infancy. And the years for tumor dormancy, everybody knows this is a hard area, and but still very cold. So that's where you probably should pay attention because it's a critical issue, but it's hard to study. But it's hard for other people, pretty often also hard for ourselves too. So we want to ask the question, if we would come into the field, can we have a better and a better chance to succeed? In this case, we do because we have ability to develop a unique dormancy model. This is something, and we and Cindy and many others working in the field using hormonal naive prostate cancer model to induce a tumor dormancy and later on the progression. I don't think I need to go into the detail, but we, we got the very first paper now under review, so hopefully can be published pretty soon. Given that, this has come to the final slide I want to share with you. So overall, we know how to dealing with proliferative aggressive cancer, and no matter the chemotherapy, radiation therapy, most of the targeted therapy, all trying to dealing with active proliferative cancer, but pretty often leave the dormant cancer behind. So if we know how to manage a dormancy, and then we how with the conventional success, and then the disease we dealing with can be there, but not so scary anymore. That's why I was thinking, and to generate a world free of cancer, probably against the mother's nature, but a world free from the fear of cancer could be possible. And that's actually, I think that's a very practical vision we could have. And I want to end with thanks to the people in the lab, Stephen Shaw and Dong, and Cindy, Connie, and Jennifer, in particular for the MCT4 project, and Hui for all the models, Fiona for working on, kind of small molecule, PK and PD, and many other things. And also I want to mention, and the HP1 Alpha project mainly carried by Xinpei and Dong. And of course, many of the collaborators are so smart, and they are so helpful. And to do whatever you want to do, you need to have funding. So I stop here. I hope you have some questions